After studying this module, you shall be able to understand why oxygen needs to be transported efficiently in living organisms, role of hemoglobin in carrying out the vital function of oxygen transport, historical evolution of hemoglobin, who discovered this magnificent iron containing metalloprotein, who elucidated its function, and finally, how was its structure discovered. Description of the structure of hemoglobin, a brief elucidation of functions of hemoglobin in transport of oxygen, carbon dioxide and nitric oxide, biosynthetic pathways involved in hemoglobin synthesis, hemoglobin disorders that occur due to inadequate supply of oxygen to vital organs. Oxygen is the most essential element responsible for sustaining all forms of life on earth. If an adequate supply of oxygen is not circulated throughout the body to vital organs and tissues, then it may lead to severe health disorders such as brain damage, organ failure, and even death. This respiratory demand for oxygen cannot be satisfied simply by the diffusion of oxygen from the environment to the respiring tissues. In fact, special transport systems are required to carry out this vital function of oxygen transport in living organisms. It is here where the role of hemoglobin, the most intensively studied protein, comes into picture. What is hemoglobin? Hemoglobin is an iron containing oxygen transport globular metalloprotein belonging to a family of heme proteins found inside the red blood cells, erythrocytes of nearly all vertebrates as well as plasma of some invertebrates. Each red blood cell contains more than 300 million hemoglobin proteins that are responsible for carrying oxygen from respiratory organs such as lungs or gills to the rest of the body, that is the tissues. So you can see over here how the hemoglobin protein is carrying oxygen from the various lungs to the tissues to the rest of the body. The transported oxygen provides energy to power important biological functions like oxidative phosphorylation of an organism. Studies reveal that the average lifespan of hemoglobin is same as that of red blood cells, that is 120 days, and approximately 6.25 gram of hemoglobin are synthesized and destroyed every day in an adult person. Besides, the normal hemoglobin levels differ between males and females, ranging from 12 to 16 grams per dl in women and 13 to 18 grams per dl in men. Now, how was this hemoglobin historically evoluted? That is the historical evolution of hemoglobin. In 1840, a German scientist named Friedrich Ludwig Hunefeld discovered the oxygen carrying protein hemoglobin by viewing the blood of an earthworm. During his examination, Sir Hunefeld reported, "I have occasionally seen in almost dried blood placed between glass plates in a desiccator rectangular crystalline structures which under the microscope had sharp edges and were bright red. These red crystalline structures were called hemoglobin." Thereafter, a series of articles describing the growth of hemoglobin crystals through successive dilution of red blood cells with solvents such as pure water, alcohol or ether, followed by slow evaporation of the solvent from the resulting protein solution, were published by a German physiologist, Otto Funke, during the year 1851. A few years later, the mechanism involving reversible oxygenation of hemoglobin was revealed by Felix Hoppe-Seyler, while the function of hemoglobin in the blood was elucidated by French physiologist Claude Bernard. However, like many other proteins, the structure of hemoglobin was too complex to be determined until X-ray crystallography made it possible. Max Perus, an Austrian-British molecular biologist, established the molecular structure of hemoglobin which was indeed a great discovery as it helped him win the Nobel Prize in 1962. Next, we should learn about the structure of hemoglobin. Therefore, structure of hemoglobin. Hemoglobin is a tetramer with a molecular weight of 65,000, consisting of 4 homologous globulin chains, which are oligomeric proteins possessing a 3-dimensional structure that are connected by disulfide bridges. The normal adult hemoglobin molecule contains 2 alpha globulin chains comprising of 141 amino acids and 2 beta globulin chains comprising of 146 amino acids. It is the arrangement and interaction of these amino acid residues present within the protein that not only governs the protein's shape but also contributes substantially to its function. In infants, the hemoglobin molecule is generally made up of 2 alpha chains and 2 gamma chains, while in adults the gamma chains are usually replaced by beta chains. Attached to each globulin chain is an iron porphyrin complex known as heme, which plays a vital role in transporting oxygen and carbon dioxide in our blood, wherein we have demonstrated that the hemoglobin molecule primarily consists of 2 alpha chains and 2 beta chains, and attached to each of these chains is basically the heme group. The heme group consists of a single embedded iron atom held in the center of a square described by 4 nitrogen atoms and exists as the heart of an array of organic rings called a porphyrin. Each hemoglobin molecule binds 4 oxygen molecules as it contains 4 iron atoms, each capable of binding 1 oxygen. It is the iron contained in hemoglobin that accounts for the distinctive red coloration of our blood. Further, details of the structure of hemoglobin, that is the conformation changes in oxygen binding, shall be discussed in the other modules. Next, we move towards the functions of hemoglobin. You know, due to low solubility of oxygen, only a fraction of the total oxygen gets dissolved in the circulatory system. However, a large amount of oxygen is needed for the conversion of foodstuffs such as lipids and carbohydrates into available energy in the form of ATP. Therefore, oxygen carrier is required for increasing oxygen carrying capacity of the circulatory fluid. Now, the protein hemoglobin serves as the oxygen carrier in blood of vertebrates and has an oxygen binding capacity of 1.34 ml oxygen per gram. In an oxygen rich environment, hemoglobin binds with oxygen with high affinity and subsequently releases oxygen in the tissues where oxygen content is not enough. In the tissues, hemoglobin picks up carbon dioxide for its transport to the lungs in a form of complex known as carboxyhemoglobin. Besides, this hemoglobin also plays important role in regulating blood pressure as it can efficiently transport nitric oxide, which can relax the blood vessels, allowing greater flow. Now, next we move towards the synthesis of hemoglobin. Now that you have a fair idea about the structure and the function, we must know how hemoglobin is synthesized. You know, the synthesis of hemoglobin takes place during the proerythroblast stage of RBC cycle by a series of biochemical reactions in the bone marrow. The heme part of the hemoglobin is synthesized in the mitochondria and the cytosol of immature red blood cells, whereas the globin protein parts are synthesized in the cytosol with the help of ribosomes. Similar to the other proteins, the synthesis of globin chain is determined by the nucleotide sequences in the structural genes on chromosomes 16 and 11. So let us get an idea about how hemoglobin is being synthesized. So we have shown that in figure. The synthesis of heme begins with the condensation of glycine and succinyl coenzyme A to form alpha-libulinic acid in the mitochondrion of the cell. So, this part of the reaction is happening in the mitochondrion of the cell. Thereafter, the ALA is transported to the cytosol, where a series of reactions generate a ring structure called coproporphyrinogen 3. This molecule then returns to the mitochondrion and undergoes an addition reaction to produce protoporphyrin 9. Protoporphyrin 9 subsequently combines with iron to form heme with the help of an enzyme known as ferrochelatase. Heme unit, thus synthesized, leaves the mitochondria and is joined to the globin chains in the cytoplasm. A disruption at any point in the production of hemoglobin may result in severe disorders such as iron deficiency anemia, lead poisoning, thalassemia and sideroblastic anemia. So, we have already demonstrated the hemoglobin synthesis in the diagram in figure, wherein we have shown how the cycle is being carried out. Next, after you have an idea about the structure, function and synthesis of hemoglobins, we will move to the hemoglobin disorders. Now, what are hemoglobin disorders? Hemoglobin disorders are inherited blood diseases that could lead to inadequate supply of oxygen to vital organs. Such disorders are passed down through families in similar way as blood type, hair color, texture, eye color and other physical traits. It has been estimated that approximately 3 lakh babies are born each year with severe form of these diseases. Generally, these disorders are most prevalent in tropical regions, but population migration has spread them to several countries. Therefore, efforts must be applied worldwide to effectively reduce such type of disorders through a strategic balance of disease management and prevention programs. We will talk about the first hemoglobin disorder, that is sickle cell anemia. Sickle cell anemia is a genetic disease of the red blood cells associated with episodes of acute illness and progressive organ damage due to abnormality in the oxygen carrying protein hemoglobin. This disease develops as a result of mutation in a gene on chromosome 11 that codes for the beta subunit of the hemoglobin. This leads to improper formation of hemoglobin molecules, causing red blood cells to be rigid and take a concave shape like sickle used to cut wheat. We have shown that in figure where you can clearly see how the red blood cells are changing their shape and in the form of a sickle cell. Because of their irregular shape, these sickle cells can't squeeze through the small blood vessels as easily as almost spherical shaped normal red blood cells. As a result, irregularly shaped cells get stuck in the blood vessels, which then stop the oxygen transport to various tissues, causing severe pain and damage to the organs. Next, thalassemia. Thalassemia is an inherited autosomal recessive blood disorder that affects a person's ability to produce hemoglobin, resulting in mild or severe anemia. It occurs when the production of hemoglobin chains, mostly the alpha and the beta forms, is largely impaired, eventually causing the destruction of red blood cells. We have shown that in figure where a brief comparison between the normal and the thalassemia cells clearly show how the red blood cells are getting malformed in case of thalassemia. Their shape is completely changing, as a result of which these red blood cells cannot function further. So, primarily there are two types of thalassemia. Firstly, AlphaThalassemia, which occurs when genes related to the alpha globin protein are missing or mutated. Normally, 4 genes are required for the synthesis of the alpha-globin chain. Moderate to severe anemia can occur when more than 2 genes are affected. In such a case, the beta-globin chains combine to generate abnormal beta tetramers that cannot bind oxygen. Then, beta thalassemia occurs when similar gene defects affect production of beta-globin protein chain. Beta-globin chains usually need 2 genes for their synthesis. Most severe form of the disease affecting both the chains. In this case, no such alpha tetramers are formed. Instead, the alpha-globin chains get degraded in the presence of beta-globin chains. Next, we talk about Porphyra. Porphyrias, derived from the ancient Greek word Porphyra, meaning purple, are a group of rare inherited metabolic blood disorders caused by altered activities of enzymes within the heme biosynthetic pathway, which was explained before, involved in the production of porphyrins and heme. In other words, it occurs due to genetic mutation at any of the enzymatic steps explained in figure. So, moving to figures, you can see how while alpha levulinic acid is being converted to porphobilinogen and subsequently porphobilinogen is being converted into uroporphyrogen by the enzyme which is called as PBG deaminase. A deficiency in this enzyme results in the accumulation of PBG derivatives primarily affecting the liver. So, any of these essential enzymes missing at any of the steps can lead to such a disorder. As the body lacks the enzymes required for completion of heme synthesis, porphyrin tends to get accumulated in the tissues and bloods, thereby affecting the skin. In such a case, it is called as cutaneous porphyria or nervous system acute porphyria. The symptoms of porphyria vary depending on which essential enzyme is missing. However, the most common symptoms of this disease include abdominal pain, light sensitivity, numbness, paralysis or mental disorders. Next, we talk about carbon monoxide poisoning. Carbon monoxide is a very dangerous gas and can cause fatal poisoning since it binds hemoglobin preferentially over oxygen when both are present in the lungs, owing to higher affinity towards hemoglobin in comparison to oxygen. Once carbon monoxide sticks to hemoglobin, forming a very bright cherry red carboxyhemoglobin, it keeps riding around, never giving their seats up to oxygen. Eventually, blood loses all of its ability to transport oxygen and there is no way to get oxygen to your brain, heart or other cells, which eventually stops all chemical reactions. So, we have described this aspect in figures where you can clearly see how on the left hand side of the diagram, hemoglobin is carrying oxygen and carbon dioxide, whereas on the right hand side, when there is carbon dioxide present in excess, hemoglobin binds to this carbon dioxide very tightly, and as a result of which the oxygen and carbon dioxide can no longer be carried out. The equation for the binding of carbon monoxide with hemoglobin has been shown in the following equation. You can see how Hb plus 4CO are combining to form HbCO4. So, inhalation of even a trace amount of carbon monoxide can cause headaches, fatigue, depression and dizziness. However, if exposure is chronic, it may lead to serious complications like heart diseases and even death in certain cases. Now, to summarize, what all you have studied in this particular module? Firstly, oxygen is the most essential element that sustains all forms of life on earth, and If an adequate supply of oxygen is not circulated throughout the body to vital organs and tissues, then it may lead to severe health disorders such as brain damage, kidney failure, organ failure, and even death. Special transport proteins such as hemoglobin are required for efficient transport of oxygen across the body. This oxygen carrying protein was discovered by Hunefeld in 1840, and its final structure was determined by Max Perutz after many years later with the help of X-ray crystallography. What is hemoglobin? Hemoglobin is a tetrameric molecule consisting of globin chains that are attached to an iron porphyrin complex called heme, responsible for binding oxygen. Apart from transporting oxygen from lungs to the tissues, hemoglobin is also involved in the transport of carbon dioxide from the tissues to the lungs and in the transport of nitric oxide for regulating blood pressure. The synthesis of hemoglobin occurs during the pro-erythroblast stage of the RBC cycle by a series of biochemical reactions in the bone marrow. Abnormalities associated with hemoglobin synthesis may lead to serious disorders such as sickle cell anemia, thalassemia, porphyria, and CO poisoning, as a result of which oxygen cannot be transported efficiently to the vital organs. Therefore, hemoglobin is a very important metalloprotein which needs a lot of emphasis and needs to be studied thoroughly